Hello everybody, welcome back to Experimental Design and Analysis. That is the Experimental Design and Analysis section of the Core Skills modules. And today, this is the 9th of 10 lectures on the sort of stats and experimental design theory, which will be assessed primarily through the exam in May, June. And we're going to look at a topic that I started last week called analysis of variance. And today we're going to kind of expand that topic, look at some important variations on a theme. To remind you what kind of experimental design or sort of analysis problem we're talking about, and also my own peculiar interests in ecology, these species on screen are demonstrating an important biological difference. They look remarkably similar, but this thing on the left is a bumblebee which can sting you. This thing on the right is a hoverfly which can't. And so I spend a lot of my time studying this phenomenon of mimicry, particularly actually amongst hoverflies. So not really relevant to the subject of experimental design and analysis, but there you go. So the outline for today's lecture, which is as usual going to be in 3 parts, is first of all to basically continue the topic that we started last week, analysis of variance, and look at what happens when you can't meet the assumptions of what is a parametric statistical test, and so instead have to reach for a non-parametric test. That test is called the Kruskal-Wallis test, and we're going to look at what it's for and how it works. And then in the second and third part, we're going to move on to more complicated analysis of variance problems where there are in fact multiple factors operating simultaneously. I'll explain what that means and the fact that it gives rise to a really important concept of a statistical interactions between factors. And so that's an important learning outcome from today, is to understand what that means. And then finally, we're going to look at the assumption of independence, which underlies most of the stats tests I've been talking about so far. We've looked at some narrow situations in which that assumption is not met, notably when we've looked at paired t-tests, for example. Today we're going to look at, briefly, another kind of problem with independence and how it can be fixed using a variation on analysis of variance called nested ANOVA. Okay, so that is the outline for the whole lecture. In part 1 then, we're going to look at the Kruskal-Wallis test. I'm sorry about the horrible name. All these stats tests have difficult to remember funny names. But in this case, what we're really talking about is a non-parametric equivalent of one-way ANOVA, or single factor ANOVA. It's not going to work, this non-parametric test, in more complicated designs. We haven't got time on this course to deal with those in a non-parametric form, but it does at least give you an alternative for the commonest form of analysis of variance when you've just got a single factor. So, reminding you, the assumptions of analysis of variance are that the data are independent, as I just mentioned, that the variance is equal, which we can test with Levene's test, and that the data are normally distributed. And if the data are not normally distributed, then we've obviously got a problem. We can try to address that problem by transforming the data mathematically, but if that still doesn't make the data normal, then we are definitely going to need a non-parametric version of ANOVA in order to make any progress. And it's that situation we're going to look at today. So it's important to note at this point that so far I've kind of said, oh, the data need to be normally distributed, and I haven't quite clarified what I meant by that, in the sense that when we looked at a t-test and I said, oh, your data need to be normal, what I meant was that, let's say you're comparing the heights of men and women, I meant that the sample that you get from the men and the sample that you get from the women must both follow a normal distribution, or approximately normal distribution, in order for the assumptions of the t-test to be met. In other words, we were thinking about each sample separately, not the whole data set pooled. So if we pooled all of the data together, there are lots of reasons why it wouldn't be normally distributed, even if the underlying samples are normally distributed. So for example, if you look at this frequency histogram here, it's a bit lumpy, but it sort of fairly obviously has 2 peaks. It's what we call a bimodal distribution. And you glance at that and you think, well, that doesn't look normal. And it isn't. If we ran a Shapiro-Wilk test on it, it would not be normal. But the reason it's not normal might be something that we completely understand in the context of the analysis. It might be that this top peak up here is a male sample, if we're looking at height, and this bottom peak is a female sample. So if you take 2 samples which have different means and you combine them into a single sample or a single pooled data set, it's very likely it's going to be bimodal if those two samples have different means to start with, which is normally what you're looking for in the statistical test. So when you have multiple groups, the assumption you're making with normality is not that the pooled data set is normal, but instead that each individual group is normal. And that's a key distinction. And in fact, in more complicated designs, it becomes effectively impossible to make that distinction very clearly. And instead, we need to rely on a sort of a slightly more hardcore statistical concept, which I'm now going to tell you about. So let's keep on with that example of we've measured height in men and women. So we've got the males on the left here and the females on the right. The mean for the men is higher than it is for the women. There's some spread around the mean in the individual data points, which are little crosses on this diagram. Both in the men and the women, you get the odd woman who's taller than the average male, you get the odd male who's shorter than the average female, but nevertheless it looks like there is a difference. The differences within each of these samples between each data point and the mean, we have previously met actually as what I think I referred to as the deviate, but we could also call it a residual. It's most commonly called residual in the context of analysis of variance. And that residual variation, that residual distance, is the bit of variation in whatever you've measured that is not explained by the factors in your analysis or the independent variables that you know about. So in our case, in my example, if this was sex and we're looking at men and women, we know that sex partly determines how tall you are, but not entirely. So some of the difference between these data points on the left and those on the right is explained by sex, but some of the variation, like this top data point here, the green one, which is quite a lot higher than the mean, is not entirely explained by sex. So this individual is very tall. Part of the reason why he's tall is that he's a man, but he's tall even for men, and so there might be other genetic or environmental factors at play for that particular individual. And what we can actually do here is partition the variation in height into the bit that is explained by the fact that he's a man, that's the difference between these two means, and the bit that's not, which is the residual or leftover variation, which is the difference between this man and the average man. And we've also called that the deviate previously. So the deviate or the residual variation is actually the thing that we assume is normally distributed. Because for any statistical model, any statistical analysis, no matter how many factors or continuous predictor variables, no matter what the independent variables look like, we are, with a parametric test, nearly always assuming that the distance between each individual data point and the prediction of the model, which in this case is represented by the mean, we're assuming that those distances are normally distributed. And the nice thing is you can calculate the residuals from any particular analysis, and it gives you just one set of data points which you're expecting to be normally distributed even when they're pulled together. Because what we're saying is that the differences between the mean for the men and this individual man, and every individual man, when combined also with those differences for the women, assuming the variances are equal, which is one of those other assumptions, those differences should all together be normally distributed. They'll be normally distributed around a mean of 0 if you think about it, because half of the data points are going to be above the mean and half of them below on average, because that's what kind of the definition of the mean really. And so if you take all of the positive residuals from the men and the women, you're going to end up with a bunch of positive data points on one side of the graph, and then you're going to get a bunch of negative ones on the negative side of the graph, and the middle, the mean difference, is going to be 0 on average. So that is actually the assumption that we make when we're running parametric tests that assume a normal distribution. Rather than worrying about having to test each sample for normality, which actually becomes effectively impossible in some complicated designs, really what we need to do is test the normality of all of the residuals from the entire data set. So that's the bits of variation which are left over when you've accounted for the things that you care about, like sex in our example. So that's what we're going to do in practice for biology students, for example, when you're running analysis of variance in the workshops. I mean, in the workbook, you're going to actually extract the residuals and test them. And for the rest of you, if you encounter analysis of variance in the wild, in the real world, that's also what you'll do. I don't know if the neuroscientists are doing it with Paul in PRISM, for example. It may be one of those things that you only encounter if you need it in your final year project, for example. Okay, so if the residuals don't follow a normal distribution in analysis of variance, then we're going to need a non-parametric test, and that test is called the Kruskal-Wallis test, which is the non-parametric equivalent of a one-way or single-factor analysis of variance. So we're going to look later on at situations where there's more than one factor, but so far, last week, for example, I just talked to you about a single factor, which in the example I worked through last week was bird species when we were looking at Darwin's finches. So if the data, or rather the residuals, are not normal, or the variances are not equal, the non-parametric test is a suitable alternative. Just like with a lot of non-parametric tests, it uses ranked data, so it takes the raw data and ranks them in order, and then runs an analysis on those, which effectively allows us to ignore the distribution of the data, the degree of spread, the variance, and also whether it's bell-shaped and normal or not. And essentially what we're looking for, just as with analysis of variance, is for variation within and among the groups that we've got in our analysis in terms of the rank. So if the data are put in rank order, we're interested in whether all the high ranked data points end up being found in one group and the low ranked in another group, or if in fact they're spread equally among the groups. So just like with all normal stats tests, we're going to compute a test statistic and a p-value to go with that. The test statistic is going to follow a known mathematical distribution which allows us to calculate the p-value. Luckily for you, the computer will do the hard work for you. All you need to know is to understand the basic framework, which is you are applying a test to calculate a statistic, to calculate a p-value in order to work out whether or not the data are likely to have occurred under the null hypothesis, or if instead you have to reject the null hypothesis because the alternative is more likely. So we're not going to look at any maths or doing this in a computer. We're just going to focus on a kind of biological example to help you remind you what the point of this exercise is. So another ecological example, I can't help being an entomologist who studies insects, so let's look at another insect example. What we've got here is a wasp, and this is a rather unfortunate and rather randy male wasp that is trying to mate with a flower. Because this is a phenomenon you really see in the natural world, there's a particular species of wasp which flies up to these orchid flowers and tries to copulate with the flower, despite the fact that it doesn't look very much like a wasp. And in the process, it collects these pollen packets on its back, which is sticky. They come off the flower and stick to its back. It then flies to another flower and tries to copulate with that and spreads the pollen to that flower, pollinates the flower as a result. So it looks like, and we're fairly confident, that the orchid has evolved to exploit the wasp here as a convenient means for moving its pollen around and pollinating other flowers. And the wasp doesn't seem to realise what's going on. But what's mystifying about this relationship from a human point of view is if you look at that flower, it doesn't look like a female wasp. And so it's a bit puzzling as to why the male is doing what it's doing. And the answer seems to lie in the fact that wasps spend an awful lot of their time worrying about what things smell like rather than what they look like. They use pheromones to communicate, and the female uses a pheromone to attract the male, and the male uses that pheromone to find the female. So the hypothesis in this experiment, given that we can't smell any pheromones of wasps with our own nose, was that there must be some kind of chemical attractant, a pheromone, a sort of fake pheromone, which is attracting the male wasps and causing them to do this dysfunctional behaviour. So we're going to look at an experiment where different types of flower or different types of stimulus were used, and the behaviour of the male wasps was observed, and specifically the copulation frequency of the males, or the amount of time spent copulating by the males, was observed when he was offered either an actual real life flower or an actual real life wasp, a female wasp, or various possible false stimuli. So there was a control, which was just a piece of plasticine or some kind of blob of stuff that was about the same size as a flower, but was completely untreated and not related to the plant in any way. So it was just a sterile piece of material. Let's call it plasticine. I can't remember what it was in the paper. And then they took that dummy female wasp, if you like, and treated it with two different chemical extracts. One was essentially just mushed up flowers. So they took the flowers and mushed them up and produced an extract which they painted onto this piece of plasticine, which then allowed them to test whether the smell, the odour of the flower was the thing that caused the male to copulate with something that otherwise clearly wasn't a female wasp. And then the final treatment was having done some chemical analysis of the orchids, they had a hypothesis about which chemical it might be that was attractive, and so they looked at that particular chemical to see if that affected specifically the behaviour of the wasps, if the males were willing to mate with the piece of plasticine if it had this particular pheromone painted onto it. And the null hypothesis in the experiment was that there'd be no difference between these 5 different treatments in the amount of time spent trying to copulate with the flower by the male wasp. So here are the data, and they're a bit messy because they have quite a lot of zeros in, which reflects where the male just did nothing. Otherwise, the number tells you how many seconds were spent during the experiment trying to copulate. And so we can calculate the averages and we can start to see some kind of pattern. So the control down here was just a piece of plasticine about the same size and shape as a female wasp, and some of the males did sort of investigate it and do a little bit of sexual behaviour with it, but on average they didn't spend much time trying to do that. Whereas with the real female wasps and the flowers and both of the artificial stimuli that were painted with the scent of a flower or with the artificial pheromone, all of those seem to have attracted more attention from the males, suggesting that our basic idea, which is that the flowers are as attractive as female wasps because they smell like them, could be right. But we need to test that formally using a statistical test to make sure that the differences we see here aren't just ones that could have arisen by chance under the null hypothesis. So the presence of all these zeros are particularly problematic because they mean that the data are not normally distributed. So if we were to do the analysis of variance, a one-way analysis of variance, and extract the residuals from that analysis, in other words, all of the bits of leftover variation in male copulation time which are not explained by membership of one of these 5 treatments, we'd find that they're not following a nice bell curve. Instead, they're rather wonky, with these zeros in particular causing the distribution to look funny. So if the Shapiro-Wilk test tells you that the data are not normal, you've got to do something else. So we'll do the non-parametric Kruskal-Wallis test, do it in a computer, don't need to worry about the maths here. It will generate a test statistic, which I've previously referred to as chi-squared, but sometimes it does get referred to as H. So you will see both written, doesn't really matter, it's just a name for the statistic. This thing here, 12.38, is the answer for this particular data set. The bigger that number, the bigger the differences between the mean ranks of the groups, which would imply a scenario that was less and less likely under the null hypothesis. So how likely under the null hypothesis? The answer is P. The computer will tell you that. In this case, it's 2%. There's a 2% chance of seeing this value of H or chi-squared under the null hypothesis. The null hypothesis says these treatments have no effect on male wasp behaviour. The male behaves the same in all 5 treatments. The chances of us seeing the data we've seen, if that's true, are about 2 in 100, so pretty slim and crucially below the cutoff value of 0.05 that we normally use as our significance threshold. The other thing the computer needed to know in order to calculate that p-value was the degrees of freedom, df, which in a Kruskal-Wallis test is just the number of groups minus 1. There were 5 groups in the experiment, 5 treatments, so degrees of freedom is 4. So when you write this up in a report, a practical report or your project, you would quote the test statistic, degrees of freedom and the p-value, and from all of that we would conclude that there is a significant difference among the treatments. So we reject the null hypothesis and conclude that the males do indeed spend different amounts of time trying to copulate with the different treatments. Great, so we've got an answer, but it's not quite the answer that we set out to get, because specifically we wanted to know, do the orchids use this kind of fake pheromone in order to attract male wasps? Which really involves us having to compare specific groups here. We want to know, if that hypothesis is correct, and it is the particular pheromone that we think it is, then all 4 of these top groups, in all 4 of those groups, the males should spend lots of time trying to copulate with the object or the female, whereas in the controls we should see little or no interest. And that is what the data suggest, but formally all we've done so far is said that there are differences somewhere in here, that these means are not all the same. So how do we square that circle? We actually specifically want to compare the control to each of these other experimental groups in a pairwise fashion in order to know whether, for example, the pheromone really does have the effect that we think it does. So, I mean, common sense gets you a long way here, because we look at the means, we can plot them on a graph, and it's clear that the control is the lowest in terms of the amount of time spent copulating, and by quite some way. So that might be enough really to support our conclusion. But if we want to go further, we could do post hoc tests, and we've already met these for analysis of variance. I'm not going to go into any more detail about them, but effectively in a non-parametric situation, they're like doing two-sample Wilcoxon tests, comparing the control, in this case, with each of the other treatments. And the reason they're special, they're post hoc tests, not just Wilcoxon tests, is that they need to be corrected to guard against the risk that if you keep on generating lots of p-values from the same data set asking the same question, you're going to end up potentially making a type 1 error more often than you want to. In other words, rejecting a null hypothesis when in fact it was just a chance event and you shouldn't have rejected it. So the post hoc tests deal with that problem. If you have this particular situation and you want to run them, then you can look up how to do it, what the best test is for your particular situation, but I'm not going to bore you with those details in these lectures. Okay, that's the end of part 1. In part 2, we're going to look at more complicated situations where instead of just a single factor, like in the previous experiment, the factor was the experimental treatment, it had 5 levels, we're going to look at situations where there's more than one factor, a rather more complicated form of analysis called two-way analysis of variance.